All right, I'm going to try to share my screen here. I think we got everybody's going to show up today. I'm sure they'll pop in if they see the email. Okay, now I got to figure out how to get your all's chat up so I can see the chat. Let's go here. All right, so welcome, welcome. Hopefully everybody still has power and will maintain power. But let's hit Lab 3 or Lab 4 tonight. I'm sure hopefully everybody got the email from the college as well as myself. I think there may just be canceling classes all week. They probably should. So the quiz is up. A couple announcements about that. The quiz is up. Let me know if you have problems. I know some people I had to reset their quiz for whatever reason, and I'm happy to do that. Just let me know. I'm trying to think what else we had. This will be the last lab until our Lab 1 exam. And also welcome a few folks outside the class. I know some people through my spotlight session, I think their lab got canceled, so I offered them to hang out with us through our Zoom meeting through the lab. So you guys are welcome back anytime. Just shoot me an email and we'll get you added on to the email list. All right, so if you can probably, yeah, no problem, Emily. I did see your email, Doc, that I saw it. Okay. Sorry, I'm not sure if this microphone. Let's talk about Brownian motion. Brownian motion basically is the definition I'm going to use. It's the motion or movement of random motion of atoms, molecules, like very small. All right, and they're just zigzagging around, and this is kind of what makes up. It's more like a physics term. It makes up matter. This is what matter does. It's just moving around everywhere. Okay, atoms, molecules, and such. And depending on the temperature, really will how high the temperature is depends on how fast these molecules or compounds move. So this is what Brownian motion is, and you want to highlight these bolded words, obviously. The random motion of molecules colliding with one another in our bodies or even in matter in general. The only time these molecules, Brownian motion, really stops is when we were to reach an absolute zero temperature, okay? That is ultra, ultra cold, and that's when all molecules cease to move at all. It's called absolute zero. You don't have to know that. I'm just trying to put it in context. Okay, so the next topic we want to discuss is called passive transport. And we've been kind of talking a little bit about this through the spotlight sessions. It's really just passive transport or simple diffusion is the same thing. It's molecules moving from an area of high concentration to an area of low concentration. And we have different parameters here that help kind of shape how fast these molecules move from high concentration to low concentration. And higher temperature, of course, will cause a faster rate of diffusion. And then if your particle size is smaller, if the particle size is smaller, it tends to move at a faster rate of diffusion. And then this kind of with molecular size and, well, with particle size and molecular weight, they kind of go hand in hand. Chances are, if you have a small particle size, you're going to have a low molecular weight. So this kind of brings me up to my third point is when you have a smaller molecular weight, you also have a faster rate of diffusion. And then obviously the example that we would use in person, that is, if the more concentrated a particle is, the faster the rate of diffusion. So if we were in lab today, we would use the methylene blue stain. And what would happen is that I would drop some of this methylene blue stain that we used last week for our cheek cells, and I would drop it into a beaker of water. And what happens is that over time, this beaker of water would fill up with this blue dye, because what's essentially happening is that we have this large concentration of stain, or high solute methylene blue stain, and we drop it into a solvent, which is the water, and then over time it will diffuse out into the beaker to where the beaker is uniform, okay, uniform color blue. I'm sure everybody who maybe noticed, or you can imagine in your heads, is that if you were to drop some food coloring or some food dye into a clear liquid such as water, you would kind of see the food coloring disperse out. And that's kind of what we're trying to talk about here. And then now if we were to put like a Bunsen burner, a hot plate underneath the temperature, and we were to boil the water, or at least heat the water up, you would increase the Brownian motion, right? So in this case, there'd be a faster rate of diffusion because the temperature is higher. Same with particle size. If each individual one of these atoms or molecules that make up the methylene blue is small in size, and you're going to find out that it diffuses a lot faster through the solvent molecules. Think of it as like a little porous lattice. A porous means, has holes, like a block of cheese has holes in it, so to speak. I'm going to say porous. That's the same thing. And then a lattice structure is kind of like just kind of like moving through a holy structure, I guess, a structure with a lot of holes. And this is how it kind of diffuses out. That's what I mean by lattice. Okay. Now I'm going to scroll down here to page 4.3, where we would do this. This is kind of what I just talked about, right? I want to note this word equilibrium because it's in bolded. Okay. Equilibrium just basically means where you have equal concentrations of dye and water. And this would be like what the end game is, right? Eventually, over time, these two concentrations would equal out, and everything would be just fine. You'd see a uniform in the speaker. Okay, facilitated diffusion. Facilitated diffusion basically is a selective permeable membrane, and this is what happens when we have. This is what happens when we have in our cells, right? This is when we have cell membrane transporters that regulate or selectively regulate what comes into the cell and what comes out. This would be most notably sodium and potassium through like an action potential, calcium, chloride, magnesium, all the different electrolytes that play a role in cell survival, all have to be regulated by these channel transporters, membrane channel transporters. And I think we're going to do an example later on about this, but this is mostly called facilitated diffusion. There's a bolded word down there that says, if the ion molecules require the aid of a membrane protein to move in and out of the cell, then it's facilitated diffusion. That's an example of facilitated diffusion would be symporter, uniporter, or antiporter. Okay, osmosis. Now, sometimes in past years, osmosis has been tough for folks to understand. And what it really is that you're talking about water, all right? And when you're talking about water, you're talking about the solvent, all right? So when you're trying to answer a question about solute, osmosis really has nothing to do with the movement of solute. That is concentration gradients or diffusion. When we're talking about osmosis, we're tracking where the solvent goes, all right? And solvent is opposite of diffusion gradients. So water moves from the side of a membrane with low salt concentration or low solute concentration to a side of high solute concentration. Now, the example I want to use here is let's say if somebody were to spill their bottled water on one of the desks in the lab, right? and all of a sudden we were to put a big salt brick right in the middle of that puddle of water. And what would happen is that the water would be consumed up into the salt brick. Okay. And what this is kind of demonstrating is that osmosis, or the water, the solvent, will go from low concentration, which is on the desk, into high salt concentration, which is found inside the salt brick. So it's like absorbing it up, so to speak. That's the best example I can kind of give. But we're going to go into what really happens when we have water imbalances in the body. Okay, so please please highlight or arrow those the definition of osmosis. It's basically how water will go from from a low salt concentration to a area of high salt concentration. Alrighty. So down here we have pictures of red blood cells. Now this is kind of interesting because this is what happens when we are to get a bag of saline or we are dehydrated, and what really happens in our body. And the red blood cells are affected by this. And I want to start off with the one in the middle, B. It's called a hypertonic solution. This basically is what happens when we become dehydrated, and almost severely dehydrated, because our bodies can take a lot. Everybody has their limits. But sometimes when we are severely dehydrated, what happens is that we are low on water. And for that case, since we are low on water found in our circulation or in our bodies, then we are subsequently have a high solute concentration, right? So you take away the water, but you leave the salt. What happens? Well, you have a high salt concentration. And what happens is that when this happens outside the red blood cells, is that it will pull water from inside the cell to outside the cell. All right, because think about it. If we're low on water and we have a high salt concentration throughout the body, it's going to start pulling more water out, and some of this water is stored in the red blood cell. So you have these cells. The cell in part B is called what's called a crenated cell. It's kind of like a shrunken up cell. I don't think you have to know that word, crenated, but if I were to see this on a slide at work, I would note this to the physician and nursing staff that this patient is likely dehydrated because they have crenated reds. Typically, a lot of elderly people struggle with their water imbalance, and they don't realize how dehydrated they really are. Okay, so now let's go over to a hypotonic solution. And this is what really happens when let's say if we drink like a lot of water at one time, maybe like a gallon of water, and it takes a while for our bodies and our kidneys to adjust to this. Sometimes our red blood cells will kind of blow up like a balloon. And that means they're just taking on so much water because there's so much of it. We are low on solute concentration, low salt in this case. And they kind of somewhat distend out. Isotonic solution is just normal. This is what happens when over here in B and A. This is what would happen when we have if you were to given a bag of saline, right? This is what 79% or 81% or 79% NaCl. All right, that is physiological saline. That is basically what our bodies are used to, and anybody can get a couple liters of this when they go to the doctor or emergency room. Okay. Now we're going to revisit those later on in the lab, but I want to kind of introduce those terms. Is there any questions? I know I've been kind of rolling through this, so I need to stop and ask if we have any questions or if we want to revisit anything. I think it's fairly simple material if you just think about it. As long as you can keep solute concentration and osmosis separate, then I think we're going to be okay. Okay. So normally, if we were in person, we would kind of do a demonstration here, but I'm going to try to explain what we're doing. And what we're really going to end up doing is talking about dialysis and how the kidneys really regulate water and the buildup of of metabolic waste. But let's go down here to filtration, down here at the very bottom of page 4 or 5. So filtration basically is the passive transport process where water and solutes move from high hydrostatic fluid pressure to areas of low hydrostatic fluid pressure. So this is how I'm going to explain what hydrostatic pressure is. The best way I can explain what hydrostatic pressure is, think of it as like when you have a washing machine and it's going on a spin cycle. Okay, when you go on a spin cycle, all of a sudden you have your clothes that are spinning at a high rate, and it pushes the water out of the drum and kind of somewhat dries your clothes. And what happens is that this is using hydrostatic. The washing machine is generating this hydrostatic pressure to push water out of the drum and into another collection area. Okay, and does everybody understand that? Can you chat me something to make sure you're following me here, or speak up? I just want to make sure I don't lose everybody, because we're about to apply. Okay, we're about to apply this to. Okay, we're about to apply this to kidney filtration and dialysis. What dialysis does. So this is all right. Cool. Thank you. This is what happens when this is how the kidneys filter blood. Okay, when kidneys filter blood, it uses hydrostatic pressure and a structure called the glomerulus. If I don't think about it, I can say it. Glomerular list. Miss. I've pronounced that better in past semesters. I'll try that later on. I'll try to pronounce it again later on. So what happens is that and these are all found in the nephron, the kidney nephron, and without going into too much detail, it basically takes the blood and puts it in this washing machine, so to speak, and will spin it really fast to push out any kind of metabolic waste to be collected within the urine. All right. So water solutes will move through the pores of the membrane and kind of down this pressure gradient. If by chance our kidneys are damaged over time, either through the one thing that can really damage the kidneys is chronic high blood pressure. If you think about it, this glomerulus, anyways, is a very delicate structure. And what happens is that if you have high pressure coming in to a delicate structure such as the glomerulus, it will damage it. Okay, and what happens is what it causes a too much filtration. Okay, there's too much filtration, and you can have a water imbalance, and albumin can get in there, and once albumin gets in certain body cavities, you look distended because it draws water in. Proteins draw water in from where they're not supposed to be. But we can save that another time. So you have to go on dialysis. It smokes your kidneys. And when it smokes your kidneys, you have to go through dialysis. And there's two types of dialysis. There is peritoneal dialysis, which is the lesser of the two. And then there's the one we always think about, which is essentially renal dialysis, where you have to go to a dialysis clinic. And what happens is that it will take your blood out of your body, somewhat try to put it in this washing machine drum, as I'm going to use this example again, and it's going to spin your blood and pull all the metabolic waste out. Because see, you have a buildup of metabolic waste, protein breakdown, which would be called BUN. BUN. I'm trying to think about it. Urea nitrogen. I'll think about what the B is in a minute. It's escaping me. It's urea nitrogen, basically, which is the breakdown of proteins, and then you also have this protein called creatinine. And creatinine is something this is what we measure when we go to they'll measure creatinine because it is such a constant protein that is excreted from your muscles. All right. It's just used protein that it no longer needs, and it will be constantly created and excreted into the urine. And it's a good measuring stick to understand how well your kidneys are moving. Okay, if you were to go to the ER and they need to give you a CT scan and they need to give you barium sulfate, which is some kind of dye that will illuminate your innards or your gastric tract, they will give you barium sulfate. However, this is a heavy molecule, and it tends to be very hard on the kidneys. So the physicians will wait to look at your creatinine to make sure that your creatinine is being filtered at a high rate or a normal rate. So if they were to give you this barium sulfate, it doesn't completely smoke your kidneys out. And this is just, creatinine is a very important measuring tool to figure out how well your kidneys are functioning. GFR also, glomerular filtration rate. That is how fast we are filtrating your blood. And basically what it does is that it uses the high concentration of metabolic waste, and it goes from high concentration to low concentration, which is the collection bag. Your urine, so to speak, will be low concentration of metabolic waste. So it helps pull this metabolic waste out of the blood, BUN, creatinine, and into the urine for micturition. Micturition is just a fancy word for urination. Okay, so are we okay there? That was kind of a roundabout way of trying to apply what BUN is, what creatinine is, and how it is filtered out of the kidneys using hydrostatic pressure. If you got any questions, let me know, but I'm going to assume that we are all together on this concept, because we're about to do a 90-degree turn. Okie dokie. Cell cycle. We talked about this morning. Cell cycle. This is mitosis, in case everybody. Okay, thank you, Paige. They're always like feedback, because it's usually I see faces in the class and I can get an idea what these facial expressions look like, and I'm just staring at a chat to see where people are. I'm not even sure if they're watching TV or if they're watching me, but thank you for that. The cell cycle is made up of four different phases. Okay, and what happens is that we have growth 1, G1, which is the preparation of gathering all these different elements to replicate DNA and to undergo mitosis. And then after G1, we have this S phase. This is where we have DNA synthesis. All right. Usually there's been some questions in the past of which phase is DNA synthesis in, and it's always going to be S phase. And then G2 is basically more growth and preparation for cell division, which is mitosis. So before we go to mitosis, we have to replicate DNA, right? We cannot make two cells without replicating the DNA to put the DNA in the new cell. So once we undergo DNA replication, we will once we undergo DNA replication, now it's time. We have 2 copies of each chromosome or each of the genes. Now we have to split up the cells. This is what the purpose of the cells is to undergo division, and that is through mitosis. Okay, so there's been questions in the past where people will try to describe what interphase is. Interphase is the other 3 phases of the cell cycle. If you look on the pie graph here, right up here, it says interphase. So that includes G1, S, and G2. This is all interphase. So don't get confused when there's a question asked you about interphase, because it's really just the boring part, or the cell replication and the growth preparation of mitosis. So once we get into mitosis, and I'm probably going to go back and maybe I'll go back and share my screen later. So when we have mitosis, we start with prophase, right? Prophase is basically the breakdown of the nuclear envelope, because if you remember, the nuclear envelope surrounds the nucleus, right? And inside the nucleus is where we have our DNA chromatin, i.e. chromosomes. And we have 23 of them, and we need to break them up. And when we have 23 chromosomes, each side of the chromosome is called a sister chromatid, right? And this is what we need to break apart. We need to break apart each sister chromatid in the middle where they connect. I made this little design this morning with my hands where this is a chromosome. They kind of like, they kind of like make like an X structure. So I'm trying to bend my hands as best I can. So this is a sister chromatid and this is sister chromatid, and they join in the middle called a centromere, okay? A centromere. And I don't want you to get that. I'm going to chat this in here. I'm going to bold it out or cap it out, a centromere. All right. This is different from centriole. We'll talk about centriole next. Folks get that confused in past semesters and they lose points. So when we have a centromere, they tend to break apart there. So let's go back to prophase. Prophase, we're breaking down a nuclear envelope, and we are kind of getting ready to spread these chromosomes out. But before we do that, we have to line them up. We have to line them up, and that is through metaphase. So there's pictures in your lab where you have these chromosomes. I used my head as the cell this morning, and I'm probably going to do it again tonight, where these chromosomes start meeting right in the middle, and they'll be vertical. They'll be down the middle of the axis of the cell. And this is kind of what metaphase is. This is the aligning of the chromosomes. Then all of a sudden we have these centrioles. This is the second term I introduced, the centrioles, and they will migrate to each pole of the cell. So if my head is the cell itself, you have a centriole over here and you have a centriole over here. And what they do is that they will start creating these mitotic spindles that go across, kind of like this. And think of them as like train tracks. And these are the cytoskeleton that's going to help migrate these chromosomes or sister chromatids apart. And I think the mitotic spindles are made up of microtubules, if we've discussed that in lecture. So now we have the alignments of the chromosomes. We have the centrioles built in with their mitotic spindles. And now anaphase, which is always the test question, is the pulling apart of the sister chromatids. All right. So anaphase is basically, there's pictures of this in the lab where we start to pull these sister chromatids apart into one layer over here on this side of the cell, and the other sister chromatids are going to be over here, right? And then we get into telophase. Telophase is just the wrapping up of the pulling apart, and we're going to restore the nuclear envelope. Okay. And so once we restore the nuclear envelope, now we have one copy of each gene in two different areas of the cell. And what we're going to do is we're going to split this cell down the middle. All right. We're going to take my head basically and just cut it in half. And that is called cytokinesis. That is where, and now if you were to split my head, now you have a cell over here and a cell over here. And that is essentially the purpose of mitosis, is cell division. Cells are here to make more cells. This is how we survive, and we replace cells before it. So it's kind of interesting in the fact that we have this huge process where we take these chromosomes and we replicate them, and then we split them apart. And it's important that's why we have G1 and G2, is that we have all the tools we need to make this happen. And eventually we will do this again, undergo DNA replication, make a copy of the gene, and then we would continue this process again. Is there any questions on mitosis? I think if you were to kind of get some pictures of this, either through the lab or you can always Google image some of these stages. Okay, thank you, Sarah. it kind of makes a lot of sense. But anaphase is always and I'm not going to say it's the test question this semester because I don't know, but in past semesters, it's always anaphase, which is, you know, the big phase where we're pulling apart the sister chromatids. Yeah. Okay. Yeah. The head visual. You gotta get creative when you're teaching from the house. Sometimes I do that. I try to use like real life situations or use my own, you know, facial features, I suppose. All right, so let me jump back into the lab. And there, it's all laid out for you through the lab, what I just mentioned. There's pictures of the lab on page 4.8. And then I want to go down to, you can draw these out in on page 4.11, and you can kind of get an idea of what these look like in an onion tip cell. All right, so let's go down. I'm going to go back to share. Sorry, I just stopped the record. All right, I pressed the wrong button here. Let's go there. All right, so I want to try to be a little bit interactive here because I want you all to think about some of these and chat the answers out, okay? Or you can chat the answers out or say them, but this is going to be basically the study sheet, okay? So the zigzag motion of particles resulting from collisions with smaller particles is called what? It's the first thing we talked about.
Brownian motion?
Yeah, that's right. It's Brownian motion. Brownian motion's that's what it is. So that's the answer to number 1. What effect would increasing temperature have on Brownian motion? Well, it's kind of like that hot plate example I used, right? If we're going to increase the temperature of the fluid, it's going to increase the amount of water or molecules moving around. So it's going to increase Brownian motion. All right, so what's diffusion? Somebody tell me or chat something what the definition of diffusion is. When we're talking about diffusion, we're talking about concentration. But it's the concentration of what? That's the answer, number 4. It is the diffusion is the movement of high concentration of solute to areas of low concentration of solute. So that's number 3. And what did I just say? I said solute. So that's number 4. Circle solute. Okay. For number 5, for molecules with a given amount of kinetic energy, small molecules move rapidly than large molecules. This difference is even more pronounced when diffusing through a semi-solid substance such as an agar. If you all ever heard of protein electrophoresis, this is how we can uniquely separate proteins that we want from other proteins that we're not interested in. This is a lab technique that uses electricity conduction to migrate proteins into an agar, and depending on how well they migrate through this agar, you can separate proteins from other proteins. Okay, so small molecules can slip through gaps more easily than large ones. So that's they're going to migrate faster because they're smaller and they can move through this lattice of solid material a lot better than larger molecules. So given the following solutions, well, these molecular weights, this kind of goes back to what we talked about in the beginning of the first page. We have potassium permanganate, methylene blue, and Congo red. Potassium permanganate looks to be the smallest of the molecular weights, so it's going to move the fastest, all right? Congo red is the largest, so it will subsequently, because it's the largest, it will move the slowest. And then methylene blue's in the middle, and it's just going to be just right, I guess. It's in the middle. It'll migrate farther than Congo red, but not as far as potassium permanganate. So that is the answer to number 5. How would temperature affect diffusion rates? Well, I probably mentioned it earlier, and I talked about absolute zero too, which is the lowest temperature you could become, and that's like, deep. There's going to be no movement at absolute zero. It's something Kelvin. I cannot remember what it is. Maybe it's 0 Kelvin. But anyways, you don't have to know that. It would be, so at high temperature, it would be an increase in diffusion rate, and then low temperature obviously slows things down, so you'd see a decrease of diffusion rates. Number 7, a blank membrane acts as a barrier to the movement of some molecules, but allows other molecules to pass through. Well, we briefly went over this, but I learned it as a semi-permeable membrane, but I think in the lab it is called a selective permeable membrane. It's the same thing. So that's the answer to number 7. All right, now number 8. Define osmosis. So what is osmosis? I'm not going to answer this one. You all can answer this one. Chat me something. Tell me what osmosis is. All right, maybe I'm getting crickets. Osmosis is the flow of solvent or water from low solute concentration to areas of high solute concentration. This is water on a table following the, moving into the salt brick, because the salt brick has high solute concentration. And when you hear water, you can also think of that as solvent. So that's the answer to number 9. Okay. We didn't do this demonstration in number 10, but you probably should revisit the red blood cells table, and this will kind of explain what isotonic, hypotonic, and hypertonic is. Hypertonic is more of a dehydrated state where water is pulled out of red blood cells because there's such a high salt concentration in the extracellular fluid. And then hypotonic basically means we have too much solvent. So a lot of, there's not enough salt to keep the water out of the red blood cells. There's nothing pulling it out of the red blood cells. So water floods into the red blood cells and kind of blows them up like a balloon. And isotonic is what its physiological saline is. It's like normal rates. Let's go down to 13 real quick, and I'll try to explain what happens here. If we have two artificial cells above, they're separated by a selectively permeable membrane and to water, but it's impermeable to sodium chloride. So that means that we're going to have 30% of sodium chloride over here and 20% of sodium chloride over here. And water is going to move based on the salt concentrations, okay? So what happens is that if it's osmosis, then this water is going to go to the area of high solute concentration. And that is 30%, which is higher. That's an A. So you're going to see water move from B to A. All right. So all of a sudden, cell A becomes hypotonic because there's more water going into A. And then B becomes hypertonic, Hyper, on B, and hypo, Hypo, hypo and the A box. If you were to become hospitalized and need to be given fluids through an IV intravenously, what would the type of fluid happen? If you were given hypertonic saline, what would happen? Well, it would essentially dehydrate you, okay? you're going to be pulling water out of the red blood cells. So you don't want hypertonic. You don't want this high salt concentration going into your extracellular space or your circulation. This is going to pull all the water out of your cells and into your circulation system. It's going to increase your blood pressure too. So you would not want that. You want isotonic saline for number 14. So you'd have a movement of fluid from inside your cells to outside your cells. That's the answer to 14. And I think that is our last question. Okay. Well, we kind of rolled through that fairly quickly, and that's kind of expected given the Zoom meeting. Do you guys have any questions? Oh, it looks like sorry guys, I just went back and I just saw the new chat. All right, let me go back to where I haven't seen this. It looks like you guys were commenting. So, okay, I didn't see those when I was sharing my screen, so I apologize. Higher temperature increases diffusion rate. Yes. And the movement of water. Yes, Sierra, it will. And then how water will go from area of low salt to high salt. Well, this goes back to chapter 2 and chapter 3, Emily, where you have these polar bonds and this attraction between positive sodium atoms and oxygen and water can somewhat be negatively charged. So you have this attraction between negative and positive dipoles that will pull water into the salt brick. That's more of like a real like general chemistry explanation where you're dealing with polar bonds and hydrogen bonds and how they kind of can pull themselves into a certain space. And then if more water, like water will fill up spaces where there is no other water. And we've probably seen this with water on our windshields where they kind of cohese together. They have this cohesion and they kind of will combine. Water does not like break apart. Water will kind of reach over to other puddles of water, and all of a sudden you have this large puddle of water when you have these little droplets. Once these droplets come in contact or near each other, they make one big droplet, and that's cohesion, surface adhesion, or surface cohesion, I think is what it is. And this is kind of the same concept there too. Water tends to track or seek out other water molecules, and they create this flow. Okay. All right, Kendra, you had a question mark on low salt concentration to high salt concentration. That is osmosis. So if you were to put salt in two different areas of water, then water is going to be attracted to the area of higher salt concentration. I'm not sure if I can email you all these answers, Paige. They are recorded, and I'm probably going to throw this up on the YouTube account so you guys can go through this again and copy them down. But yeah, there's no answer sheet like we had in Lab 1. All right, so since I got all this chat back up, did I answer all your questions now that I see the chat? Are we okay here? Because this is it. This is all of Lab 1, or this is all of Unit 1. Okay. All right, cool. Well, we made it through. I'm surprised my electricity hasn't been cut off yet. I'm just waiting for that to happen because I was about to go buy a generator this weekend. I'm like, no, I don't want to spend the money on it. I'm not sure if some of you all are old enough as I am, if you remember 2009 and 2002, when we had really bad ice storms. I think I was out of power. Our folks were out of power for 8 days, and it sucked. And I'm not doing that ever again. So if I want to go shell out the money for, yeah, you were a baby. Yeah, I'm 31. So I was probably 2009. I was a sophomore in high school, maybe a freshman. maybe a no 2000 no excuse me 2009. I was in college. I was at Murray. I came home because they were shipping. It was so. If anybody knows where Murray State is, it's in the far western area of the state by Paducah, and they brought the National Guard in to bring kids down to Tennessee Martin. I said, "The hell with that! I'm driving home." And I didn't see lights until I got to Versailles. It was a 3.5 hour drive, 3 hours and like 45 minutes without any lights. In 2002, that was the worst one. That was the 8 days with no electricity. I was a seventh grader. I was a seventh grader. Okay. So hopefully we'll get through all this and everybody will be safe. If you have any questions, let me know. If there's anything that pops up on the email or any questions that pop after, email me, and I will put this up on YouTube so you guys can go over this again. Last comment is that this is the last bit of Unit 1 for the exam. So I think next week I think we're off because of President's Day, and it's kind of like a preparation period for exam 1. We will be doing Proctio on this, so I'll have to send out a how-to protocol on getting Proctio uploaded into your computers, so you can be monitored while you're taking your exam. So unless you guys have any other questions, I'm going to call it for tonight. And for some of you guys that were visiting us, you're more welcome to come back at any time. Just let me know, and we will accommodate you. So I think other than that, we're okay. We're good to go. I'm going to cut this